Okay, this is the blood chapter, first chapter we're going to be covering this semester. About physical characteristics. First of all, the normal pH level of blood is 7.35 to 7.45. I would have that tattooed on my ankle because you're going to need to know that for a long time. Normal body volume is about 5 liters. We're going to cover that again in a few more slides. Give or take a little more depending on body size, about 5 liters of blood. It's maintained by different hormones. Hormones such as antidiuretic and aldosterone help maintain blood volume and therefore blood pressure. And atyl natriuretic peptide decreases blood volume. So functions, blood transports primarily any substance that can be dissolved in plasma, and essentially which is water, can be mobilized. And so we are transporting gases such as oxygen and carbon dioxide, nutrients that are being transported from where they're being absorbed in the stomach and the small intestine, to the liver and to other, to skeletal muscle, hormones, which are chemical compounds produced by glands to target distant organs and their cells, enzymes. Water disperses heat really well, and different waste products. As a consequence of cellular metabolism, all those waste products have to be removed, and so it'll be transported in blood. Maintains homeostasis through the pH buffers, where how we maintain our 7.35 to 7.45 normal pH range, body temperature, and it acts in a protective mechanism as well. We have platelets that are active in blood clotting, and then we have our white blood cells that are active in disease resistance. So the first thing you want to think about when it comes to blood is you have two major divisions. You have plasma and you have formed elements. Plasma, is normally about 55% of the volume of blood, or given like a microliter of blood, 55% should be plasma, and about 45% should be your formed elements. Formed elements are your red blood cells, your white blood cells, and your platelets, erythrocytes, leukocytes, and thrombocytes. Erythrocytes will always be far more numerous, about 94%, and a little more 4 to 5% would be platelets, and then less than 1% is generally about the number of white blood cells, so the percentage of white blood cells. If you were able to take a little capillary tube and get a droplet of blood and separate and centrifuge it down, or if you had a little vacutainer and you could centrifuge it down, once you would see where your plasma is and where your erythrocytes are. And you can even just kind of eyeball it in that respect. You should have a little more than half of the volume of blood to be more of that straw colored plasma to the top. And then the red blood cells would be to the bottom. So you should always have a little bit more watery substance than you do all your formed elements. All right, so here let's you see the most abundant cell you're looking at here are your erythrocytes. White blood cells are easy to identify, but there are 5 different classes, and we're going to talk about that. They are much larger. They have nuclei that generally stain either purple or abundant, like orangey granules within the cell. Platelets are kind of little dots. If you could take a purple magic marker and just kind of pop, 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 a slide, that's what a little platelet would look like. So they're much, much smaller than your erythrocytes and smaller than your white blood cells. Okay, so plasma. Plasma is about 90% water. Some books will say 92%, but about 90%. And so the water component, acts as a solvent and suspends different, not only other substances that need to be transported, but the formed elements, and then it helps transport gases. We have plasma proteins that are about 8%. They remain in the blood and do not enter the cells. They're generally produced by the liver. Collectively, they maintain an osmotic pressure to regulate blood volume or maintains an osmotic gradient in blood, which means it helps draw water to it. Albumin is one of those. It's the most abundant to maintain an osmotic gradient. Fibrinogen is active in clotting, and gamma globulins are active in the immune response. They're the antibodies. And then about 2% are your gases, electrolytes, nutrients, wastes, enzymes, and hormones that are being transported. All right, so this little slide, this is from your textbook, just talks about the same things, just in a different form. 90% water. You have lots of electrolytes, and over the course of this semester, we'll talk a lot about different electrolytes, and then one of the last chapters, we kind of pull it all together a little bit. But the plasma proteins have big functions, and the albumin, and the gamma globulins, that are formed by the liver, are really important because, formed elements and these like albumin and these proteins should stay in the blood, and at a capillary level, that becomes important because we lose some fluid at the capillary level to the interstitial spaces, but then we need to kind of draw it back in on the venous side of a capillary bed to help maintain blood volume and therefore blood pressure. And so the liver's ability to form all these proteins, is a vital function. And if the liver isn't working well and forming these proteins, or maybe you have kidney disease and you're losing some of these proteins in urine, then you're going to have problems with maintaining the appropriate blood volume and blood pressure. All right, so this just talks about gamma globulins, fibrinogen, and some of those other substances that are transported in plasma. Okay, so hematopoiesis, right, is the formation of our blood cells, red blood cells specifically. All blood cells are formed in red bone marrow. Remember in AMP1 talked about spongy bone, said that there are two major components of spongy bone. You had trabeculae and red bone marrow. And red bone marrow forms all blood cells. Well, all blood cells, right, red blood cells, white blood cells, platelets, come from your red bone marrow. So hematopoiesis, occurs in red bone marrow. All blood cell types arise from different stem cells. And once cell begins to differentiate, it cannot change. Hematopoiesis is the formation of blood cells. Erythropoiesis, right, is the production of red blood cells. Normally, you produce as many red blood cells that are going to be dying off, so to speak. So production equals the rate of destruction. And normally that's about 1.5% of your red blood cells are going to probably be worn out and removed from circulation. They only, circulate for about 100 to 120 days, depending on whether you're male and female. And so therefore a certain number are always going to be becoming very fragile and starting to be broken down in the spleen and being removed, so that you're always kind of starting to mature some erythrocytes to take the place of those that are dying off, so you can maintain a normal, stable volume of cells. The stimulus to increase production is generally hypoxia or low blood oxygen levels. The hormone is called erythropoietin, referred to as EPO, and it's produced by the kidneys. And then the erythropoietin will travel to the red bone marrow and stimulate it to begin production of maturing more red blood cells. During one of its last maturation phases, the nucleus is ejected from the red blood cell, so it becomes what's called anuclear. So it's one of the few body cells that doesn't have a nucleus. That's your red blood cell. And at that stage, it's called a reticulocyte. One of the advantages of losing the nucleus is that you develop this biconcave shape to it. So it kind of looks like a lifesaver candy. And so it dips into the interior. And that gives the cell a lot of flexibility, to pass through capillaries in small spaces, to be able to bend a little bit and then pop back up again. So here it shows you the negative feedback loop for hematopoiesis. Start with low oxygen delivery, low oxygen levels, or hypoxia, may be due to a decreased number of red blood cells. It could be that you have an adequate number of red blood cells, but they haven't matured correctly, or perhaps we're not producing enough of them, or perhaps the hemoglobin is faulty, and not able to transport oxygen adequately. So there are many different reasons or etiologies. It could be from blood loss, that it was due to hemorrhage. But in any event, the oxygen levels are low. You secrete erythropoietin from the kidneys. It targets the red bone marrow. Red bone marrow starts putting out into circulation more red blood cells that mature and ultimately transport more oxygen, and therefore increase your oxygen level in the blood. So erythrocytes, they originate in the red bone marrow. They have a biconcave shape to it because they are anuclear. They live for about 120 days, anywhere from about 100, 110 to generally about 120. They contain hemoglobin, which is a quaternary protein structure. Remember, proteins, especially quaternary, tertiary, and secondary structures of proteins are maintain that shape because of the hydrogen bonds that are created from the polar covalent bonds within the amino acid chain itself. And because they are maintained by hydrogen bonds, they are susceptible to high temperature and low pH. And when those conditions are present, then those hydrogen bonds start to break down, and now your hemoglobin starts to unravel, and it can no longer transport the hemi group, which has the iron molecule in the center, which is what bonds to the oxygen. So the fact that it has these large proteins within the red blood cell that function to transport the oxygen, then it's really important that proper blood pH is maintained in that normal range of 7.35 to 7.45 and normal body temperature, so that we don't denature the proteins within our red blood cells and start to decrease our oxygen carrying capacity. Red blood cell counts reflect the blood's oxygen carrying capacity. So the number and quality of red blood cells will determine how much oxygen you are able to transport. A little bit of oxygen, a very small amount, is actually dissolved as a gas in plasma. A very small amount. So the way you're going to be transporting that is based on the integrity of the erythrocytes that you have in circulation. And functionally, they transport oxygen, carbon dioxide. Typical ranges for about a microliter of blood is about plus or minus 5 million. You can see that for the males, the females, and the children. Pretty much the same number of red blood cells per microliter of blood. And that the normal range for an adult, and that's given the size of the adult as well, but the average adult size is about 5 liters of blood in total. So changing the amino acid sequence. This is what happens, or what produces sickle cell anemia, is when you have in the 6th position of your amino acid chain that codes for making hemoglobin. When we have a substitution of valine for glutamine, right? We substitute in here a valine. That then tells the cell when it's making its hemoglobin to make it in sort of a faulty form. And what happens is that when you're under a low oxygen tension environment, the hemoglobin and the some of the proteins in the plasma membrane of your red blood cells start to sickle and form kind of a boomerang kind of a shape to it. And when that happens, you're not, or you are unable to carry the same amount of oxygen as you normally would in a normal red blood cell. And also the plasma membrane becomes very sticky, and it has a tendency to kind of clump in small capillary areas around joints in different organs, joints like your elbows and the knees, around the hips, sometimes the femoral acetabular joint, but it can also kind of form these little clots, in organs as well. So sickle cell anemia, is an example of inadequate or improper hemoglobin manufacturing. It's not like the number of red blood cells is diminished, but that the hemoglobin in most of those, are going to be faulty. The characteristics of red blood cells. Well, the hemoglobin, as I said, is a quaternary structure. So over to the right, you can see these 4 quaternary structures, or 1 quaternary structure, because there's 4 within it. You can see this hemigroup to the inside, and then the red dot represents the iron molecule, and that's what the oxygen will bind to. You generally have 2 beta chains and 2 alpha chains. That's how we kind of distinguish them. What else is important? 1 hemoglobin binds with 4 oxygens, because it's a quaternary structure. So each kind of knot there has its own hemigroup, an iron molecule and an oxygen molecule. Some carbon dioxide is transported by the hemoglobin from the tissues to the lungs as well. And then it says in the bottom, if protein is the primary constituent, what is it vulnerable to? That's what I was referring to, as far as the hydrogen bonds. It's very vulnerable to high temperature and low pH. This just shows you some pictures of the maturation process of the red blood cell or the erythrocyte. All blood cells, whether they're white blood cells or red blood cells, they start out really large and they get smaller as they mature. I mean, we're so used to us as individuals or humans, or even really any other animal, they're born very small and they get large as they get, more mature. Well, these cells, they start out very large, kind of blastic forms it's called, and then they get smaller as they mature. And then to the right-hand side, where we eject or liberate the nucleus from our red blood cell, that forms a reticulocyte, and then it further modifies to the mature erythrocyte that you see to the far right-hand side. Red blood cell production and its control. Well, it's oxygen levels, in blood that will stimulate the release of erythropoietin, from the kidneys. That targets the red bone marrow and increases red blood cell production, and therefore increases oxygen level capacities. Development and the maturation process needs vitamins and different minerals. So iron is one of them, because you need to have the iron, that bind to the hemigroup, of the hemoglobin that will ultimately transport the oxygen. But we need B vitamins as well. We need B12, niacin, and B9 folate for red blood cell maturation. And when those are deficient, then the red blood cells don't become as small and assume the shape that they should, that biconcave shape. To absorb B12, you need to have intrinsic factor. An intrinsic factor is sort of an enzyme hormone, that is secreted and produced by cells in the stomach. So their job, or one of their jobs, is to produce intrinsic factor. And when intrinsic factor is present, as long as you're taking in and you're ingesting nutritionally B12, the B12 will be absorbed and it'll be transported through the body. And then it'll stimulate the further maturation of your red blood cells. Without intrinsic factor, then you won't have the same maturation process. A CBC and a CBC with a differential. So a CBC means it's a complete blood count, which quantitates all the cells of blood and calculates other indices as well. So a complete blood count, if you've never seen or really looked at, let's say, the results of a CBC with a differential of your own, maybe of a parent or a sibling, or even your pets, you can get copies of the blood work that gets sent in for them. When you have a CBC done, it quantifies how many red blood cells and the percentage, how many platelets, what percentage of white blood cells, and then it breaks down of all your white blood cells or your leukocytes, breaks it down into 5 categories by percentage and quantity, of the 5 white blood cells that are in circulation. So it gives you the quantities and percentages of your white blood cells, your red blood cells, and your platelets. It will also give you the amount of hemoglobin. It will give you a hematocrit, which is your packed cell volume, which is your red blood cells. It gives you the average hematocrit for a male or female, which is about 45%. Females can often, be around 40 to 45, and males as well. But if you remember 45, it'll help you later when you get into nursing when it comes to the rules of 3, and that will help you kind of keep some of these indices in line. When you have higher than normal number of red blood cells, it's called polycythemia. When you have fewer than normal number of red blood cells, either the number of the cells or the quality of the cells, then it's called anemia. White blood cell counts, or CBCs with differentials. So this is a procedure to count the number of white blood cells per cubic milliliter of the blood. Generally, it's normal to have 5 to 10,000, 5,000, 10,000 per cubic millimeter of blood. Leukopenia is a lower than normal number of white blood cells. Leukocytosis is a higher than normal number. So when you look at that 5,000 to 10,000 range, you have less than 5,000, it's called leukopenia. And if you have more than 10,000, it's called leukocytosis. Now, sometimes that can be normal, especially if you're fighting an infection. If you've participated in very vigorous exercise, especially if you're like a marathon runner, or you do a lot of kind of long bike rides, the 100-mile bike rides and things like that, and or if you're very dehydrated, then it can sort of temporarily allow the red blood cells, or the white blood cells, to be higher than normal. Generally, if you are dehydrated, if you just rehydrate, those values will come back in the normal ranges. And neutropenia means that you have a count less than 4,000. Okay. Yeah, that's a redundant slide. So somehow I had that slide twice. Let me just skip over that. Red blood cell production. I'm not going to get into extraordinary amount of detail with this, but you do have to understand the basics of what is happening when you're breaking down your red blood cells. Generally, it's the spleen, I should say, with the reticular fibers there that are part of the reticular endothelial system, and they're lined with a lot of macrophages, which we're going to talk about when we talk about our white blood cells. These white blood cells help grab onto the worn out red blood cells and break them down. And when we break down the red blood cells, we're going to recycle as much as we can from it. A lot of those amino acids in the hemoglobin, we're going to try to reuse that and break it down and make it available not only in the liver, but in red bone marrow, where you're forming more red blood cells. The iron molecules, the same way. We want to capture that, and we want to transport it with transferrin, to the liver and to red bone marrow, again, where you're manufacturing, more red blood cells. But the hemi group is something we want to get rid of. So we start in the spleen, and we start breaking down those red blood cells and put it in a form that can be transported through plasma from the spleen to the liver. And we take that form of that hemi group, and we break it down into what's called unconjugated bilirubin. In that form, it can then be transported in plasma, because we have venous blood leaving the spleen. It's going to be transported to the liver next. And so it goes to the liver, and now the liver can have cells there that are going to take that unconjugated bilirubin and form conjugated bilirubin. And now that conjugated bilirubin can be put in bile, which is going to be stored and concentrated in the gallbladder, that green bulbous organ in the inferior aspect of the liver in this diagram, and then it can be transported through the duct system into the pancreas and into the small intestine, where we can eliminate the majority of it out in feces. So it becomes captured in the spleen and broken down into bilirubin, and it's an unconjugated form. Then it gets transported to the liver, where it gets conjugated into bile, where it will pass into the small intestine, and the majority of which will pass right into feces and be eliminated. And that's actually what gives feces the brown color of feces. Some of that may be reabsorbed and is reabsorbed actually while it's passing through the small intestine, and it gets essentially transported into blood and can pass through the kidneys before it goes back to the liver again. And some of that will then spill over into urine, and that's really what gives urine the yellowish kind of tint to it. So it gets broken down in the spleen. Unconjugated bilirubin is transported to the liver, where it becomes conjugated, most of which will be in bile, be transported into the small intestine, right, and eliminated in feces. A little bit ends up being absorbed into the blood and transported over to the kidneys, where it becomes eliminated. Bilirubin and the buildup of bilirubin and not being able to evacuate it adequately, or if it builds up too quickly, is what produces jaundice. Jaundice is the yellowing of the skin, of the mucosa of the eye, of the gums. You can often see that, even in the fingertips. Right, anemia is a lower than normal number of red blood cells. It can technically be a number or just a quality of normal red blood cells. We said anemia, at the end of this, I give you some examples of anemia, and there are many different types of anemia. Anemia is a very, very broad category, but it's generally due to, a couple major etiologies or causes, either underproduction, loss, like in hemorrhage, or inadequate production. Let's say, you're not, you have something like sickle cell anemia where you're making hemoglobin, but it's faulty hemoglobin. Or we have things like thalassemia minor and thalassemia major, which are other hemoglobin malfunction issues. Or it could be that you're not absorbing the vitamins that you need to, or you're not getting the vitamins, let's say, in your diet, or absorbing them, and you're not maturing your red blood cells adequately. Polycythemia is a higher than normal number of red blood cells. So different disorders, here are some of the anemias, and these they do like to talk about in nursing quite a bit, is pernicious anemia. This is an autoimmune disease that destroys some of the stomach mucosa that produces the intrinsic factor. Remember I said that intrinsic factor is made by cells that are found in the stomach and lining the stomach. And if they're not making the intrinsic factor, then you are not going to absorb B12. No matter how much you take in your diet, you will never absorb it. Right? So you're going to have either, some kind of an injection, generally, to introduce the B12 into your blood. If you are not getting enough in your diet, then you're going to have the same presentation of red blood cells, but that will be called a macrocytic anemia. And that's the difference between pernicious anemia and macrocytic anemia. The red blood cells look the same, really, but they're just due to different things. If you actually have this autoimmune disorder and you don't have any intrinsic factor, regardless of the amount of B12 you take in your diet, you're going to have these larger than normal number of red blood cells, and it's going to be considered pernicious anemia. If it's due to dietary factors, then we call it just a macrocytic anemia. And one of the reasons why you can, or ways in which you can tell the difference, is that first of all, there's a test called the Schilling's test, and that helps you differentiate pernicious anemia versus just a macrocytic anemia, like a dietary insufficiency. But another rather quick way is just to try to introduce more B12 into the person's diet. And if you do that and then you retest after about a month to 2 months, then you will generally see red blood cells that are of normal size. Here's a list of different types of anemias that are, fairly frequently occurring and common, as far as especially covering in the nursing programs. You have aplastic anemia. And that can be due to damaged bone marrow in some way, shape, or form. Aplastic anemia is common in people who are getting radiation therapy to broad areas, and especially in adults, flat bones. A lot of the flat bones and the epiphyses of our long bones, there is where we have our red bone marrow that's forming a lot of forming the red blood cells in our body circulation. And when you're having radiation therapy, it's killing off not only cancer cells, but it's killing off everything else there. So sometimes you'll have obliteration of a good amounts of red bone marrow in areas that are being treated, and it can cause aplastic anemia. And that's not that difficult to actually diagnose, because if you're blowing out and you're kind of reducing the red bone marrow, you generally see a drop in the normal quantities of all blood cells across the line, all your formed elements, not just your red blood cells. You'll see a drop in your white blood cells and your platelets as well. Hemolytic anemia, hemolytic anemia is when a significant number of red blood cells are being destroyed. And that, again, is kind of a broad category because you don't always know exactly why. It could be due to severe burns, superficial burns over significant parts of the body. Again, it'll increase the temperature. It can cause denaturation of the hemoglobin. The cells start to break down. They hit the spleen. The spleen, is taking them out of circulation. You might even have that accompanied with, jaundice, because you have a lot of red blood cells being broken down faster than the bilirubin can be eliminated by the body. It could be due to blood loss. It could be due to a transfusion mismatch, where your own antibodies are killing off and attaching to the red blood cells, that were improperly infused. Iron deficiency anemia is another common one. Again, that's generally a dietary insufficiency. So changes to the diet will often see that, change, especially for women in reproductive years and menstruating. It's not uncommon for women who are in reproductive mode and menstruating to have iron deficiency anemia, periodically due to a regular bleeding cycle. We have pernicious anemia, which is the inability to absorb the B12 because of the lack of intrinsic factor. Sickle cell anemia is the one we described earlier, where we have a substitution of valine for glutamine at the 6th position. And then we have thalassemia, minor and major. There's 2 different types, but similar. That is also, again, a defective gene, that the hemoglobin is not being made properly. A1C, you're going to see this a lot in different programs as well, whether you're in nursing, or you're going into PA school, or even becoming like a med tech, not a medical assistant, but a med tech working in the labs. The A1C test is a blood test that provides information about the person's average levels of blood glucose, also called blood sugars, over the past 3 months. And the A1C test is sometimes called the hemoglobin A1C, or the HB, because that means hemoglobin, HbA1c, or the glycohemoglobin test. And it'll often be referred to as the glycohemoglobin test. So a certain amount of glucose is stored in the red blood cells. And an A1C test is going to make a measurement of what's in these red blood cells. So the A1C test is a primary test used for diabetes management and diabetes research, and to see how well, like monitoring the patient's response to whatever therapy you're currently using. So the A1C is based on the attachment of glucose to hemoglobin and the protein in the red blood cells that carries oxygen. The red blood cells are constantly forming and dying, as I described before, right? They live for about, 120 days. For females, it's less. And there are always a few more being brought down the pike, as reticulocytes, about 1.5%, that are going to be maturing to replace those that are dying off. So since cells live about 3 months, then the A1C test reflects the average amount of glucose over the past 3 months. It's reported as a percentage, and a normal A1C is below 5.7%. It's a really good test, especially when, whenever you're monitoring anybody with diabetes, to see how well, they're either sticking to their diet, or how, even if they are eating a proper diet and medicating appropriately, either as a diabetes DM1 patient, diabetes mellitus 1 patient, or diabetes mellitus 2 patient, the A1C still gives you a really good idea of what they've really been doing. Sometimes, patients, I'm not going to say that they lie to you, but, they want to kind of eat what they want to eat instead of getting rid of a lot of sugars and things in their diet, which makes it easier for your body to maintain appropriate glucose levels. But sometimes it's no fault of their own. They're just maybe the medication, they're not responding well to the medication, or they need to have some other medication prescribed along with it. So, it just gives you at least it just tells you very objectively what the level has been in the last 3 months. And as long as everybody's, the patient is eating what they should be eating and medicating as they should be medicating, then it gives the practitioner, an idea of what they need to do. Either the current treatment plan is just fine, or whether it needs to be modified. And sometimes you need to just counsel the patient a little bit more about how important it is to take medications appropriately, what kind of diet they really should be consuming, and try to limit certain things. If there are some people, they just love desserts and processed sugar, and carbohydrates and percent, so, if that can be squelched and managed, then you usually see a better response, to the current medications they're on, and their A1C levels generally drop down. Resistance and blood dynamics. So blood resistance, or resistance, what we're referring to here, is just the opposition to blood flow. And it's sort of like the friction of blood in the blood vessels, that slows it down. It's just like rolling a ball across the floor. It won't keep rolling forever. There is friction. There's the force of gravity pulling on the ball. And as the ball rolls across the floor, you have two surfaces that are, not are roughened, so to speak, and it slows the ball down. Right? Eventually, the ball is going to come to a stop unless it's rolling downhill. Right? So if it's a flat surface, that given gravity pulling down on the ball and the ball rolling across the floor, is that frictional coefficient there is going to slow down the ball, and the ball is going to come to a rest. Well, without pumping and pushing the blood through the blood vessels, that too will come to a rest as well. So it's important to understand what forces actually work in opposition to blood flow, and then it starts to make a little bit more sense, especially later when we start looking at blood pressure. So what does it depend on? The diameter of the blood vessels that it's passing through. It's a lot easier, and there's less resistance if blood is moving through a very large tube system than one that's very, very narrow. Right? You try to push the same amount of blood through something narrow, it's going to resist it. Right? So diameter, the larger the diameter, the lower the resistance. The smaller the diameter, the higher the resistance. The length of the blood vessel. The shorter the distance the blood has to move through through a vessel, the less opportunity to slow down and to develop a friction against the inner blood vessel wall. And so if it only has to travel 3 millimeters is very different than having to travel 10 millimeters. So the longer the vessel, the more resistance and opposition to blood flow. The shorter, the better, right? The less opportunity. So viscosity is another variable. This is the thickness and the stickiness of your blood, right? The thicker it is, the harder it is to move. Think of it this way: if you ever squeeze out honey out of a little squeeze bottle, it's especially if it's been in the refrigerator, it's much harder to squeeze out of that bottle. But if you had water in a little squeeze bottle like that, you turn it upside down, you squeeze it, and water comes spurting out. You go to squeeze the honey, and it's like you gotta, put some muscle into it sometimes, to get it to come out. So that's what we're referring to with viscosity. So some of the things that affect viscosity. So if viscosity, some of the factors that affect viscosity are the number of red blood cells, right? And because the number of red blood cells, or the percentage of red blood cells, is higher than any other formed element. So if we're about 94 to 95% red blood cells, in a microliter of blood, let's say, if we dramatically change the number of red blood cells, then we're no longer 55% plasma and 45% formed elements. We might be 55% formed elements and only 45% plasma. All right, so now that blood has become a lot thicker, just to the fact that we have, let's say, extraordinary polycythemia. But we could also, maybe the number of red blood cells really is normal. Right? However, our hydration level has changed. We've become extremely dehydrated. Well, if plasma should be 55% of your blood and 90% of the plasma should be water, and we don't have a lot of water available, then again, it kind of, due to the different reasons, but it produces the same effect. Now, because we're so dehydrated, it's not because we made, we're producing too many red blood cells. It's just that we're so dehydrated, but it still produces maybe that same 55% formed elements and only 45% plasma. It still produces a thick blood, but just for different etiologies or different causes. So as we have 1, 2, 3, 4, as viscosity increases, blood flow decreases. So anything that increases the viscosity and increases the thickness of blood will decrease the blood flow. If we increase the number of red blood cells, let's say in polycythemia, then we decrease blood flow. Why? Because if we increase the red blood cells, we're increasing the viscosity. If we decrease blood flow, plasma, as in, let's say we are dehydrated and we don't have as much water, we decrease our plasma, we decrease our blood flow, because when we decrease our plasma, we increase the viscosity. And then this kind of refers to a phenomena of the beginning versus early stages and then latter stages of polycythemia. Is that generally, it's normal to start increasing red blood cell production if we have a decreased amount of oxygen being circulated through the body. And initially, when we start making more red blood cells, we start increasing the oxygen carrying capacity, but it hasn't affected the viscosity of the blood yet. We're just, oh, good, now we're delivering more oxygen. But if we don't stop producing extra red blood cells and we keep producing more and more and more, sometimes in the case of, blood doping and things in sports, but it can also happen for other reasons as well. It wouldn't necessarily taking shots of erythropoietin. Is that as you keep increasing the number of red blood cells, now the blood becomes thicker and the flow slows down, and you decrease your blood flow, and therefore you decrease your oxygen carrying capacity. So initially, it's working in your favor. You have more red blood cells and you're delivering oxygen. But then you kind of get over the hump there and you're like, whoa, well, now we have too many red blood cells and the blood isn't moving as quickly, and flowing as adequately. And now you're delivering less oxygen. Leukopoiesis is an increase of production in response to an infection. when it comes to our 5 classes of white blood cells, we have what's called colony stimulating factors. These are hormones that trigger leukopoiesis. That's all. So leukocyte structure. We have a nuclei and different organelles, just like any other cell. They are classified as either granulocytes or agranulocytes. The granulocytes, your mnemonic is going to be BEN, basophil, eosinophil, neutrophil. The agranulocytes are the monocytes and the lymphocytes. So we're going to take a look at them, and we're going to make some general descriptions, not too general, some of that kind of specific as to their appearance, so you can recognize them, on slides histologically for lab class. So if you understand and you know what are the basic kind of characteristics of these different white blood cells, it's easier for you to recognize them physically. And also on some of the models in lab, you can kind of key together, okay, I see these orange granules. I know that's an eosinophil, EO for orange granules. And then you have to commit to memory some of their functions. But these are their basic appearances. And the eosinophil generally has kind of light orangey rose colored granules, peachy colored maybe. We use a different stain to really identify the eosinophils compared to the other white blood cells. But you have eosinophils, you have basophils, neutrophils, lymphocytes, and monocytes. So we're going to take each one individually in just a moment. Leukocyte function. Leukocytes have certain capabilities. They can either, white blood cells, some of them stay in the blood and some of them are able to leave the blood and actually like chase down bacteria and viruses. And some of them will just get transported and take up kind of residence in lymph nodes and other organs, of the body. And they wait there and they sit there and they monitor for different microbes to invade the body and detect their presence. So they're specialized to aid in immunity. They're able to leave blood vessels called emigration or diapedesis. They follow chemical trails, or they're capable of following chemical trails called chemotaxis, and they can increase production, upon infection. And what's interesting, or what you need to know about each class of white blood cells, is that different things stimulate them to increase in number. And so when you know what stimulates which white blood cell to increase in number, then you go, oh, well, now I know that I have a viral infection because my lymphocytes are elevated, or my neutrophil count is really high. There's probably a bacterial infection. And if the neutrophils are high, but the monocytes are not high yet, it's probably an early infection. And if the neutrophils are high and your monocytes are high, then it's probably an infection that's been going on for a little while. Okay, so our cells contain what's called major histocompatibilities antigens on their cell surfaces. And so all our nucleated cells, especially, let's say, all my nucleated cells in my body have the same MHC complex, this chemical compound found on the surface of the cell. And that way, my white blood cells who are looking for foreign invaders don't start to respond to my own body cells. So as long as I have this chemical compound on my body on the surface of my normal body cells, is that my white blood cells won't attack it. But if you don't have that major histocompatibility complex on the surface of the cell, that's how your white blood cells know it's a foreign invader. And they start to mount an immune response to them. So those MHC complexes, there are different types. There's MHC1s and MHC2s. We'll talk about that a little bit when we get to immunity. But that's how your cells, your body knows whether those are normal body cells or not. They can't see them. The only way they can respond to them is chemically, whether something is a normal cell or an abnormal cell. Okay, so neutrophils. Neutrophils are the most numerous white blood cell normally in circulation. Anywhere, I'd say I just stick to around 55 to about 65 or 64%, 62%, somewhere around in there. It will fluctuate depending on how many are being kind of liberated into circulation at that point in time. They have, they are called a polymorphonuclear cell, which means that their nucleus twists off into little lobules and different, kind of looks like sausage links all piled together. So rather than being like a nice round nucleus, like what we looked at the cell model in the cell chapter, I think chapter 3, in AMP1, this nucleus isn't going to look like that. It's going to kind of look like sausage links. The cytoplasm is generally light lavender kind of a color, and you may see in a certain, I wouldn't, there are granules there, but it's not as abundant as in a basophil when we see a basophil. So they're often, the nucleus is multi-lobed to about 3 to 5 lobes. They function in phagocytosis. They are, and that's why they're so abundant. And they're not these smalls, but they're rather small white blood cells. They can ingest a bacteria before they kind of die off, but they can also ingest fungi. They're the first to arrive and to elevate in a bacterial infection, so that's good to know. If you see them elevated, then you know that you have a bacterial infection going on, and that they're also called a polymorphonuclear leukocyte. So they have that multi-lobed appearance there. Then we have eosinophils. So we say EO orange for kind of orangey, reddish, pinkish granules, and that's A to the right-hand side. So you have a bilobed nucleus, which is true, but the eosinophils, the only one that's going to have pale staining and abundant granules in its cytoplasm. Ding, ding, ding. And the models in lab, will look the same way. The granules are going to be orange. They produce antihistamines. They will elevate when you have parasitic infections. And normally you have about 1 to 3%. 1 to 3% of your white blood cells will be eosinophils. Then we have basophils. Right, basophils are the most infrequently occurring. It's normal to have like 0 or 1% are going to be basophils. Either absolutely none that you'll see in 100 cells, white blood cells that you count, or maybe one will be a basophil. When you're doing a microscopic evaluation of blood, it's kind of easy to confuse a basophil with a lymphocyte, and I'll show you that in just a minute. With a good microscope, you can't confuse it, but in the ones, not that the microscopes we have here at St. Pete College aren't good, but you really need oil immersion, and we don't generally use oil immersion in the AMP2. You start using oil immersion when you get to microbiology. But in any event, the basophil is very dark purple staining. It has an abundance of dark staining granules that obscure your ability to see the nucleus. So sometimes you can see the nucleus in the background a little bit, but it's not uncommon to not be able to see any nucleus. And all it appears to be is a big ink blot, so to speak, of dark purple staining stuff. If you can magnify it, and sometimes the cell will just turn in such a way you can see all these granules, and it lacks any kind of significant light lavender cytoplasm around the circumference of the cell, or maybe off to one side, then you could be pretty certain that it's a basophil. They contain granules that contain histamine and heparin. Therefore, you're going to see a lot of these associated with allergies and increasing inflammation. Histamine increases blood vessel leakiness, and heparin acts as an anticoagulant. So when in doubt, if you see a cell and you think it's a basophil, more than likely it's a lymphocyte, because that's the second most commonly frequently occurring white blood cell. But it could be a basophil. You just have to be very careful and make sure you really magnify and you really do careful inspection of the basophil before you start calling a bunch of lymphocytes basophils, because that's having an elevated number of basophils, is that kind of number is cause for concern. Let's put it that way. Right, lymphocytes, that's the next cell. Right, this is the second most numerous, about 25 to 35%, somewhere around in there will be your lymphocytes. Right? You can see this lymphocyte right in the center here. It has a very large dark staining nucleus. However, you should see some light violet cytoplasm either around the entire circumference of the cell or maybe off to one side, kind of like a crescent moon kind of an appearance. So when you magnify the cell, you won't see an abundance of dark purple granules, and you'll see some light lavender cytoplasm off to one side or around the entire circumference of it. And more than likely, what you're looking at is a lymphocyte, because neutrophils are most numerous, and then you have lymphocytes. So more than likely, it's a lymphocyte. So lymphocytes primarily respond to viruses. We have T lymphocytes and B lymphocytes. T lymphocytes are active in producing cytolytic enzymes and granules, which means that it's these granules and enzymes are going to break down, physically break down a virus, versus B lymphocytes will morph into what's called a plasma cell, and they make antibodies specifically to a particular virus. So we'll get into a little bit more of the specificity of their functions when we get to immunology. But right now you can just know that lymphocytes, we have T lymphocytes and B lymphocytes. T lymphocytes produce the cytolytic granules, and the B lymphocytes produce the antibodies. And then we'll look at a little bit more about how those work when we get to the immunology. All right, then we have monocytes. And monocytes, I call them the big mothers because they're large, they're very large. So over here on the right, you can see here we have a monocyte. And a monocyte that's left and kind of diapedetically left the cardiovascular system and is out in interstitial spaces or taking up occupancy in maybe the spleen or in lymph nodes, then we call it a macrophage. It's a very large cell. If you took about 4 or 5 red blood cells, you could stuff it into one big monocyte. The nucleus takes up, I would say, a good 80 to 90%. Well, that's a little too much. I would say around 70 to 60% of the total volume of a white blood cell. It doesn't always look like that at the cell turns, and you will see that there's a good amount of light violet cytoplasm. And sometimes if it turns in such a way, you will see a very U-shaped nucleus present. So we say, you gave me monocyte. If you see a large white blood cell and the nucleus is in a U shape, then it's a monocyte. It is capable of phagocytosis, and they're sort of garbage collectors. They phagocytize bacteria, viruses. They help break down those red blood cells in the spleen. They help break down other worn out cells and their organelles. They travel to areas where, let's say, you have a fractured bone, trying to reunite and then scar tissue over, so to speak. You're going to have macrophages there. So they activate the lymphocytes to initiate immune responses as well. Monocytes are very good at what's called being an antigen presenting cell. When they phagocytize viruses, they'll take a portion of that virus, the chemical compound, on the surface of that virus, and they will bring it to the lymphocytes so that the T lymphocytes and B lymphocytes can start to develop a specific immune response to a particular virus. And that's what we call, the specificity that the lymphocytes have. They will also respond to bacterial infections, but they will elevate a few days after the neutrophils start to elevate and respond. So therefore, once you see those monocytes, elevating higher than normal, then it's a better indication of a chronic bacterial infection rather than a very recent or early bacterial infection. Then we have platelets. Platelets are called thrombocytes. They are actually fragments, the kind of fragments of a cell that it kind of exfoliate off the surface of these megakaryocytes, which are these very large cells, the platelet cells essentially, that produce the platelets on the surface, and it kind of fragments and clusters up. Did I have a good picture? I thought I had it. I do. I have farther down. I'll show you what that looks like. So it's an anucleated fragment of a larger cell called the megakaryocyte. It's essential for clotting processes because they will stick together and form a plug. Thrombopoietin is the hormone produced by the liver that regulates platelet production. It triggers the formation of these megakaryocytes, and megakaryocytes produce about 2,000 to 3,000 of these little fragments. So down here, this gives you a better idea of what we mean by exfoliating off the surface, is that we start with this stem cell, which is a blast cell, and then it becomes a megakaryocyte, and then a stage 2 through the megakaryocyte, and then the stage 4 here is a mature form. And you see on the surface, little bits of it just start to kind of peel off and exfoliate, kind of like dry skin on the surface of your epidermis. And that's where your platelets come from. Hemostasis is how we control and stabilize blood volume, especially in the case of bleeding. So there are a few different stages. There are 5 stages of hemostasis. When we have some kind of cut, it starts with a vascular spasm. So this kind of makes sense. If you have a laceration and a cut on the surface of your body, let's say, is that you don't want to keep delivering the same amount of blood to where it's just going to bleed out to the surface of the body, let's say. So what you want to do is vasoconstrict and, produce a reduction of blood flow to that area so you don't just keep losing it, and bleeding it away. So it starts with a vascular spasm. Then we have that platelet plug formation. Those platelets will come together, and where you have a break in the blood vessel, they'll start to form, one big kind of clot, and it'll just form a plug and a knot so that it covers the surface, and blood can no longer escape the blood vessel at that point. Then we will have coagulation, which is really the blood clot, not just the plug that will occur. We have 2 types of coagulation. You have an intrinsic pathway and an extrinsic pathway. In the case of a laceration, this would be an example of the extrinsic pathway of clotting. Then we have clot retraction and repair. This is where the platelets now release chemicals that stimulate the smooth muscle to repair. And what it does is it kind of takes the 2 walls of the blood vessel, and approximates them. And it starts pulling them together so it can heal across there. And the smooth muscle can develop a kind of a continuum, in what's called the tunica media. We'll look at the structure of blood vessels, the 3 layers of a blood vessel, but the middle layers where we have all that smooth muscle. And we need to kind of bring the edges together of the most inner layer called the tunica interna, which is your squamous cell epithelium. And then you need to bring the smooth muscle together so that, kind of bridge the gap, so to speak. And now it becomes one continuous vessel. Then we have fibrinolysis, and that's where we break down the clot that has formed there in some of that plug. And you do that with what's called plasma. So you don't want to keep clotting because that could be a problem. You just want to clot, you have that platelet plug and clot just enough, to discourage any blood loss. And then you want to modify it and let it heal. And so you use fibrinolysis to break down the residual parts there so we don't clot too much and block off the blood vessel. And plasmin is what does that. So here we're going to talk a little bit about coagulation. We said that I sort of described an intrinsic versus extrinsic pathway. So blood coagulation is triggered by cellular damage and blood contact with foreign surfaces. So it can be actual laceration, let's say, of a blood vessel, or when blood comes in contact with unusual substances or surfaces, then a blood clot can form. This is a hemostatic mechanism, causes the formation of a blood clot via a series of reactions, which activates the next step in a cascade of reactions. And it can occur extrinsically or intrinsically. So you have two different etiologies. We have the extrinsic pathway, which is kind of the shortest. And I like the example of a laceration. Let's say you have, you actually cut and you essentially tore into the endothelium, which is the inner layer of a blood vessel. So you've cut through the outer, the 3 layers of a vessel. And now when your heart's beating and pushing blood, through the cardiovascular system, now you have a pathway for the blood to leave this hose system and not stay contained within the hose or the blood vessel. So it begins with some kind of endothelial injury. And then you have to have what's called the tissue factor, and we use what's called factor 7 to start this pathway of coagulation. So it's a very brief pathway before it joins right through the center, which with what's called a common pathway. An intrinsic pathway uses factor 12, factor 9, factor 11, and factor 8 to start a unique series of a cascade of chemical reactions before it too joins right here in the center at factor 10 in what's called the common pathway of coagulation. So the difference really is the reason, the stimulus for coagulation. If it's an extrinsic pathway, a good example of that is some kind of a laceration or a cut, with something like, let's say, a knife or even a paper cut. The intrinsic pathway is often stimulated by the blood passing across the surface of something that seems to be unusual or foreign. And risks of that are, let's say, when you have let's say, fat deposit in some of your blood vessels, or if you had a valve replacement in the heart. Whenever you have something foreign in the body or that creates more turbulence, then you can stimulate what's called this intrinsic pathway to form these clots. They both join in the center here with factor 10 as the common pathway, and then they follow the same series of cascade of reactions until finally, just like we did before, we're going to release plasmin and break down those clots. All right, so extrinsic versus intrinsic clotting mechanisms. Extrinsic clotting mechanisms triggered when blood contacts a damaged blood vessel wall, the endothelium. The anticoagulant in this pathway is coumadin. The intrinsic pathway is triggered when blood contacts a surface and exposes collagen. So when it's coming across, an unusual or foreign surface, so to speak, chemicals inside the blood vessels trigger blood coagulation. The anticoagulant in this pathway is heparin. These two things are really good to know. Extrinsic pathway, think of a laceration and a break in the blood vessel. Coumadin is your anticoagulant. In the intrinsic pathway, it's when blood comes across foreign surfaces, where we're triggering the coagulation because of unusual surface contact. Your anticoagulant is going to be heparin. This intrinsic pathway is why if you're going to take a blood sample and you want to test these two pathways, because you can do that. You can say there are certain time elements associated with the clotting that should occur stimulating the intrinsic pathway versus the extrinsic pathway. And when you get a blood sample for that, the needle, when you're extracting blood, and the vacutainer, which stores all the blood, has to be lined by an anticoagulant so that we don't start the intrinsic or extrinsic pathway prematurely. We want to preserve the blood in a pure form, and then when we get it down to the laboratory, then we're going to put it through an analyzer. We're going to stimulate both of those pathways and time exactly how long it takes to form a clot stimulating the intrinsic pathway versus extrinsic pathway. It's rather interesting. So both of these pathways, they end in the common pathway. A stable clot is formed, and then plasmin, again, digests that blood clot, digests the blood clot. Factors affecting clot formation are levels of vitamin K. We need to have adequate amounts of vitamin K. The anticoagulants are heparin and coumadin. The AKA for coumadin is warfarin. Remember, the heparin can also be produced by basophils, and basophil can, mast cells type of a basophil, essentially the same thing. And that aspirin can also, of course, it's not formed in the body, but you can take it orally, acts as an anticoagulant and inhibits platelet clumping. A thrombus is an abnormal clot, and an embolism is essentially a thrombus that has broken free. An embolism is a clot moving through the blood vessels and has become mobile. It could be fat, it could be another substance, it could be an air bubble, just something that can essentially take up space in the blood vessel and block it. All right, then we come to blood groups and transfusions, antigens and antibodies. So this is our blood typing information that we need. And then we're about done with this chapter. So terms to be familiar with: agglutination. Agglutination is the clumping of red blood cells in response to a reaction between an antibody and an antigen. So it's that clumping, kind of clotting, a granular looking reaction in blood. Antigen is the chemical compound found on the cell surfaces, right? They may be capable of stimulating the production of antibodies in one way, shape, or form. An antigen is one of those MHC complexes I described, how we have this normal, like the chemical compound on all my nucleated blood cells is the same imprint, the same compound found on all my cells. And that is called an antigen. Right? Foreign cells will have different antigens than mine, than my MHC complex. And therefore, a foreign cell and the antigen on its cell surface can stimulate an antibody response, let's say, in my body, if it got in my body. Antibodies are these proteins that attach to specific antigens. So antibodies are what are automatically made in the liver, as part of the antibodies, like the anti-A's and the anti-B antibodies that we have. They're made by the liver, and that's really what we're looking at here. Yes, the B lymphocytes can become plasma cells and they make antibodies, but they make those to a specific virus. These are antibodies right now when we're talking about blood typing, are ones we automatically make at about 4 to 6 months of age, once our livers after birth are really fully functioning. Right? So antibodies are specific to bind to a specific antigen, like a specific key goes into a specific lock and doesn't, they can't be mixed. Well, I guess they can technically, but we want them to be specific, one key to one lock, period. So when the ABO grouping is an international grouping based on the antigens, the first few antigens found on a cell surface. And this is how we start by identifying blood types. There are about 400 different antigens that can be found on the cell surface, but the ones that we mostly match, most easily match up to, at least initially, right, is the ABO, Rh positive, Rh negative blood types. So this is based on the presence or absence of two major antigens on the red blood cell surfaces that determine whether you're AB, or O. And then when you're Rh positive or Rh negative, that's something else, but we're going to get to that. All right, so we either have an A antigen on the cell surface, a B antigen on the cell surface, both A's and B's on the cell surface, or neither A or B's on the cell surface. So blood type A, if we have A antigens on the cell surface, we call it blood type A. And if we are blood type A, we will make B antibodies, period. If we have a B antigen on the cell surface, then we're blood type B. and we will automatically make A antibodies. If we have both A and B antigens on the cell surface, then we call that being blood type AB, and we will make neither A nor B antibodies. And you'll see why in just a moment. And if we have neither A nor B antigens on the cell surface, that's what we call blood type O. And if we're blood type O, then we make both A and B antibodies and put those out in our plasma. All right, so this picture gives you a good indication. Let's look at blood type AB. So these are just symbols for A and B antigens. So the A, we have A antigens and B antigens. The A antigens are like little triangles, and the B's are like little balls on the surface of that red blood cell. And when we have A and B antigens on the cell surface, over to the next column, it said plasma antibodies that you're going to make are the agglutinins. You make neither A nor B antibodies. Okay. Then let's go to the next one. Blood type B means you have B antigens alone on the cell surface. So we have those little green balls on the cell surface. So that individual will automatically make anti-A antibodies. Now this is going to make sense because let's look at blood type A and look at the shapes of A antibodies and B antibodies, and why, if you are blood type B, you're not going to make B antibodies. So blood type A, we call it A because we have A antigens on the cell surface, those little triangles. And look at the shape of the anti-Bs. Right? Anti-Bs, B antibodies cannot bind with a B antigen, right? They're just not, an A antigen is just not compatible. A antibodies could bind to an A antigen. So you see the ones on top, we have those purpley kind of A antibodies. You can see how those would be able to bind to an A antigen. And my battery is running down. This says it's charging, but it says I need to turn over my battery. So I'm hoping I can get through this. I'm just going to kind of talk through the rest of this rather quickly. A person who has A antigens on the cell surface that is blood type A, if you made A antibodies, then they would bind to the antigens on your own red blood cell surfaces, and you'd be killing off your own red blood cells. And vice versa, if you were a B blood type and you had B antigens on the cell surface and you made B antibodies, those would be capable of binding to the B antigens, and you kill off your own red blood cells. So you normally do not make antibodies to your own blood cells. Right? You don't make antibodies to antigens that you normally find on your own blood cells. And then here we have blood type O. You have neither A nor B antigen on cell surface, so you are totally free to form A and B and A antibodies and B antibodies because you have neither A nor B antigen on the cell surface to bind to. Okay, so we need you to know what constitutes being blood type A, B, AB, or O. Do you have A antigens or B antigens? And know which antibodies you're automatically going to make. You can review, which blood transfusion, if I was blood type A, who can I donate to? Or you can also go the opposite way and go, look, if I'm blood type A, who can I receive, blood from? I think I put a video up there that's good to review and to hold on to when you get into nursing, because that's going to be very helpful. But I'm not going to ask you questions about that. When it comes to the RH blood grouping, okay, we have the RH antigen or D antigen is what determines whether you're RH positive or RH negative. And this is not something, if you are RH positive, it means you have the RH antigen or D antigen on your cell surface. And if you're RH negative, it means you don't have it, you lack it, right? You're negative for it. If you have that RH or D antigen on your cell surface, you will never make the RH antibodies, or what you call them, D antibodies, because you don't make antibodies normally to your own body cells. Right? So The RH group was named for the rhesus monkey. RH positive means that you have the presence, or you have that RH antigen or D antigen on your red blood cell membrane. RH negative means you lack that antigen. It is more common to be RH positive, 85%, and it dominates over being RH negative. The problem is when you have an RH negative mother carrying an RH positive fetus. The initial exposure, if you are an RH negative mom and you're not taking antibody therapy or ROGAM, what's called, then if you have a mixing of blood between the fetus and the mom, which will generally happen if no sooner than at least childbirth, then you'll get a mixing of the fetal blood with the mom, and she will start to make antibodies to the RH positive blood that she got exposed to. And generally the first pregnancy isn't the problem, it's the second one. So if you become pregnant again and again you have an RH positive fetus, the antibodies that mom is making, that particular class of antibodies, is small enough to get across the placenta. And so it can start to, it can and it will kill off the red blood cells in the fetus that you're carrying. There are, before you initially, you ever get exposed and stimulate mom to make those RH antibodies or D antibodies, you can go on an immunoglobulin therapy called ROGAM to prevent her from ever recognizing the presence of some of those red blood cells should they get across the placenta somehow. And that's not uncommon, especially if you're doing amniocentesis. If there's trauma, again, vaginally birthing or, during the birth process, regardless, you can have a mixing of the blood. So this will keep mom from ever getting stimulated. So knowing your blood type before planning a pregnancy, especially if you're RH negative, is a very smart thing to do. Not that there aren't, even if she does get stimulated, there are things you can do. There are different types of umbilical transfusions and then transfusions to mom to try to interact and bind with those antibodies she might be making. But it's a lot easier to prevent her from ever making those antibodies than to try to manage it later. And there are ways where you can measure how many antibodies she's making, whether she has any at all, and what the level is. And then further down here, these, if you put this PowerPoint and download it and put it in a slideshow mode like this, when you click on these different links I have here, these different games, you'll be able to practice on your own. It gives you a score, but it's just a game to identify your different white blood cells and your red blood cells and your platelets. A couple of different kinds. And then here, the FYI notes for those of you who want to know more specifically the pathway of unconjugated and conjugated bilirubin, and what percentages are going to pass through into feces, what percentage gets reabsorbed into the bloodstream, and then from there, how much goes to the kidneys, how much is going to go right back to the liver and follow that bile pathway right back to the small intestine. This is for you, but I won't ask you about that. But I will say that, it's good to have an understanding of the difference between unconjugated and conjugated bilirubin and where, what is its ultimate fate. Here, jaundice is talking about the buildup of bilirubin. Once it exceeds a certain amount in the blood, you start to get that yellowing of the skin, the mucous membranes, the mucosa of the mouth. It's generally categorized into three different, kind of like anemia, three different etiologies. It's either considered prehepatic, hepatic, or posthepatic. Prehepatic means it's generally in the hemolytic category, which means is that it's coming from many red blood cells, higher than normal number, being broken down very rapidly and is overwhelming essentially the spleen and ultimately the liver to break it down properly. Right. So the pathology is occurring prior to ever getting to the liver's job to break it down. Hepatic, or what's called hepatocellular, is the pathology is generally located within the liver itself, where you have maybe